Today, I will discuss the process of DNA replication in prokaryotes, and I will explain the concept and enzymes that are involved in a prokaryotic replication fork. DNA, which is the genetic makeup of organisms, is made up of a sugar, phosphate, and a base that are held together by phosphodiester bonds. Prokaryotes have a circular DNA molecule, and to replicate the cells go through a process called theta replication, which is not the only mode of replication for prokaryotes, but it is the most common and can be seen in prokaryotes such as E. coli. Here, I have a diagram of the theta replication bubble. This diagram shows some terms that are important parts of theta replication. The origin of replication is where the replication starts. Prokaryotes are unique because they cannot have multiple origins of replication like eukaryotes can. DNA replication occurs only from the 5' to 3' ends, which are labeled here. The remainder of these labeled terms will be explained in the next diagram, which breaks down this process and takes a more in-depth look. Now I will discuss this diagram, which I have made using common snack foods from around my house. To begin, these spaghetti noodles represent DNA strands. The upper strand is the leading strand, which is synthesized from the 5' end to the 3' end continuously. The bottom strand is the lagging strand, which is replicated discontinuously. Now let's look at the key players in this replication process. This goldfish located at the mouth of the replication fork represents the enzyme helicase, which separates the hydrogen bonds, allowing the DNA strands to be unwound. These chocolate chips on the leading strand represent single-stranded binding proteins, which bind to the single strands and stabilize them as replication proceeds so that the strands stay separated. The pretzel here represents the enzyme gyrase, which is an enzyme that relaxes the supercoiling of the DNA and relieves pressure. The cheese puffs represent the RNA primase, which holds primers that are represented here by these chocolate Rice Krispies. Right next to the RNA primase is the DNA polymerase III, which is represented by a Mickey head goldfish. The DNA polymerase III binds to the primer at the primase and adds nucleotides to replicate the DNA strand. As mentioned earlier, this is done continuously on the leading strand, which is the upper strand in this case, but on the lagging strand, this process cannot occur continuously. This leads to the formation of small segments called Okazaki fragments, which are shown here. After the DNA polymerase III does its job, then DNA polymerase I moves in and removes the RNA primers so that it can then replace them with DNA. I have represented the DNA polymerase I with a cashew, and it would come in and remove these RNA primers and then fill in their spots with DNA. The final step in this process is up to the enzyme called ligase, which is represented by this popcorn. Ligase attaches DNA to phosphodiester bonds to completely seal the newly formed strand. In this next display, I have hypothesized the effect of a dysfunctional DNA polymerase I and what effect it would have on replication. If the DNA polymerase I can no longer perform its 5' to 3' exonuclease function, it will likely result in the Okazaki fragments being unable to connect. Also, the DNA would not connect to phosphodiester bonds. This could also result in some RNA being left over, since it would not be able to get removed. Here are the external sources I used to create this presentation.